My name is Peter Bodsky. I'm a regional marketing manager for Thermo Fisher Scientific, and I cover the ICSP business unit products, including ion chromatography and sample preparation. We're going to be talking about the superior sensitivity of haloacetic acids using a new technology called two-dimensional ion chromatography. So before I go into the innovation part itself, I'd like to talk about the history of innovation that Dionex has had over the years. And this starts back in 1975 when we first introduced our first IC system. And this arrow is actually narrowed down quite a bit, and so you can see that since 1975 there have been a host of innovations and modifications to the system, but over the last 15 years we've actually seen some great improvements in the overall system functionality, performance, and ease of use. And a few of these are listed here. Many of them have actually received R&D 100 awards. We've also seen some industrial design awards and innovation awards. So just wanted to point that out before we got started. So what are disinfection byproducts? It's really, disinfection is a treatment that's necessary to eliminate waterborne diseases that can cause contamination from microorganisms. The two main sources are from ozonation and chlorination. Ozonation yielding bromate, chlorination, of course, chloramines, including chlorite and chlorate. Now, these are highly regulated due to the nature of their impact on our health. That would include the nervous system. For chlorite, bromate, of course, is carcinogenic. And chlorate has gastroenteritis and blood disease implications as well. There's also trihalomethanes as well as haloacetic acids. And these are all now regulated under the Safe Drinking Water Act. And we're very excited to talk about two-dimensional IC in the context of haloacetic acids and how this is now going to be promulgated into the states via the EPA. So here's a summary of some of the methods that we're using actually for chlorite and chlorate. As you can see, they're largely conductivity based, and they are using a mixture of carbonate mobile phases or hydroxide mobile phases. And then we have some application notes listed here as well for reference purposes. But both can be are found actually under the EPA 300.0 Part B as well as 300.1 Part B. There's a lot of bromate methods. There's several technologies associated with bromate, and bromate is a complex analyte to quantitate. So we have sort of different technologies that we use to go after bromate. Primarily, it's suppressed conductivity, and we also have a 2D-IC method for bromate as well. In addition, there's technologies around post-column addition as well as the addition of acidified potassium iodide to C-bromate. And then finally, there's also IC and ICP-MS. All these methods actually have EPA methods associated with them. They all have their strengths and weaknesses, but I will point out that the 2D-IC method, method 302, is actually EPA approved, and we're going to talk about that extension relative to haloacetic acids. So haloacetic acids are essentially formed when chlorine or other disinfectants react with naturally occurring organic materials. And you can see the breakdown here of the haloacetic acids that we're going to be talking about. About 13% of these breakdown products are direct haloacetic acids. The rest are considered from trihalomethanes, about 20% or so. And there's a whole other category that aren't necessarily monitored, but are considered disinfection byproduct breakdown components, if you will. And these are considered unknown halogenated organics. The haloacetic acids that are a concern are the HAA5, and they're listed here, essentially monochloroacetic acid through dichloro, trichloro, monobromo, and dibromoacetic acid. And you can see the formulas there as well as their relative boiling points. So the HAA5 are the ones that are a primary concern. There are 9 haloacetic acids that are monitored, but the 5 are the ones that are typically regulated. So back in the 70s, trihalomethanes were coming to light, And we had early regulations around trihalomethanes. And then finally, in 1998, the EPA released a stage 1 disinfection byproduct rule. And this involved several new regulations, including monitoring of these HAA5s as well as bromate. And so any water municipal plant that is doing disinfection with chlorine needed to begin monitoring the HAA5s. And they needed to report the total of the 5 haloacetic acids. And we're also looking for an average or annual average of this total reported at no more than 0.060 milligrams per liter. And so, and also the DCAA component of that would not necessarily be present at all. In 2006, Stage 2 revised that rule so that we are again looking at that average, that 0.060 milligrams per liter, but they reduced the overall limits to ensure that those limits were being, further pushed down, and again, still the DCAA would not be present. So if we look at the summary of the methodologies associated with haloacetic acids, there's actually quite a good list of them. The first one is actually a GC ECD method, and it's actually probably the most popular one given that it's been around the longest, and it also has EPA methodology 552.2 as well as 0.3. The second one, 557, is actually an ICMS method, which can actually employ single quad or triple quad mass spec technology, and that's actually currently released under EPA method 557. The other one that we're going to be talking about that is pending through the EPA is going to be probably an extension of method 302 or method 314.2, and this is 2D-IC with suppressed conductivity. And you can see the relative detection limits associated with each of those technologies, including the GC, the ICMS, and now the 2D-IC method as well. So let's talk about 552.3 in the context of how it performs. It's actually a great method, and it's been around for the longest time, as I mentioned. It does, however, have an extensive workflow associated with it. This means that once you get your sample, you have to treat the sample immediately, and you can see here we're adding 100 milligrams of granular ammonium chloride to convert this residual 3-chlorine to a combined chlorine total. The next steps are really where the workflow becomes more complicated. It's actually about a 3 or 4 hour process to actually complete one sample derivatization, and in any one of those steps, if you make a mistake or the sample is not properly treated, you need to start out completely over. So it's really very labor intensive, which in fact makes it one of the disadvantages that we see relative to this method. The method actually has great advantages. I mean, it's great selectivity, it's got low MDLs, it has great applicability in terms of the concentration ranges that you're dealing with. But again, it requires this extensive sample pre-treatment. It's time consuming, it's labor intensive, and this means that it has the potential for error, and we've all encountered this as well. So here you can see the detection limits are being listed, and the percent recoveries. And so this is, again, is a well-established method. The HAA9 are well characterized and well understood, but it does tend to be quite labor intensive. EPA 557 is the suppressed ion chromatography method with MS or MS. And again, this has advantages in the detection of haloacetic acids in that it's a direct detection technology. It does eliminate that liquid-liquid extraction component, and it also eliminates coelution because mass spec is such a selective detector. You can actually really be sure that the peaks you're quantitating are actually what they are. It's fully automated, and recoveries on this is actually good. It's also 90%. The limitation, of course, has always been the purchase of a triple quad mass spec. This is a big investment, and most laboratories aren't necessarily equipped or want to have that obligation to go out and buy such a large invested piece of equipment, including a triple quad mass spec. Okay, let's shift gears a little bit and talk about what is an ion chromatography system and what does it look like. Most of you are familiar with this, but Over the next few slides, I thought it would be instructive to show sort of how this is all put together in the context of a 2D-IC system. If we look at an ion chromatography system in a single dimension format, you can see that it comprises of water or a mobile phase, which is going into the high pressure non-metallic pump. This then goes into our eluent generator, which really does bring a lot of ease and innovation to the table. And that then goes into the separation column, the suppressor, the detector, as well as in the data management system. So this would be considered technically a one-dimensional system. And in a two-dimensional system, what we're doing is we're essentially having a one ion chromatography system and following it by another ion chromatography system. So it's literally two ICs in series. In that first dimension, we're actually going to do a large loop injection onto a large loop that's associated with the 4mm system. And that 4mm system will then actually go through a separation and then put the effluent of that separation onto a concentrator column. That concentrator column will then go on to another separator column in this second dimension system, and we'll actually have a complete unique separation. A couple of fun things happen when you go from one dimension to another. You eliminate matrix, and if you reduce the diameter of the second column, or the second dimension column, you actually improve sensitivity. What does that look like chromatographically? So let's say we go from 4mm over to 2mm in the second dimension. So 4mm in the first dimension, 2mm in the second dimension. We now see an improvement by 4x in the overall sensitivity of the analyte. That's great because as we go into that second dimension, we want better sensitivity. We've also eliminated the matrix so that we can now see much better, more of the analyte without the associated matrix effects. What happens if we take this one step further? Let's say we go from 4mm in the first dimension over to 0.4mm in the second dimension. We actually see a 100-fold increase in the sensitivity. And this is good news for anyone that's doing low-level detections because it means that we can now really start to push the limits of detection in that second dimension without all the associated matrix effects. The first dimension takes care of the matrix. We're concentrating the analytes on a concentrator, doing the separation again in that second dimension, which means we can now see our analytes in the absence of these groundwater matrix effects. So it's really a phenomenal technology, and we're quite excited that the EPA is really getting close to releasing this. Just to re-emphasize, in the first dimension, we're actually doing this large loop component, which is a 4mm injection typically. In that 4mm separation, we're doing a matrix elimination. We're knocking out the matrix and trying to focus the analytes of interest. So that concentrator, that second dimension concentrated sample is going on to the second dimension separation, and we're actually done doing the separation. So this combined effect, this first dimension, second dimension separation, allows us to enhance sensitivity, improve selectivity, eliminate the matrix, and we can actually all do that under one packaged system. So it doesn't involve hyphenated techniques like IC or MS. There's no post-column addition. It's all on one complete system. So I will also mention that the 2D concept is not new. This is actually a current method under EPA 302 for bromate, as well as 314.2 for perchlorate. So this is a widely accepted and well-utilized technique. So we are seeing pending approval for this on haloacetic acids and exploring the possibility of doing this also with hexavalent chromium. You do get a really nice sensitivity improvement with this two-dimensional concept. RFIC is really critical to this in that when you're using hydroxide mobile phases that are suppressed to water, you get this lower background conductivity, so your analytes have a true, much better signal, really higher sensitivity. So using an RFIC system, again, to emphasize, when you go from a two-dimensional system, 4mm down to 2mm, there's a 4x improvement in sensitivity. When you go from a 4mm down to a 0.4mm system, you have that 100x improvement in sensitivity. So that dimension shift, in addition to the RFIC capability really enhances the overall sensitivity of the system. And so it really is a nice technology. We're very excited about this, and we anticipate that it's going to be a great success for the haloacetic acid determination. Here's a picture actually of the system, what it looks like. This is an ICS5000 Plus. It's a dual channel IC, so the first dimension and the second dimension are housed in the ICS5000 Plus. It does have continuous operation up to 5,000 psi, high pressure capable of course, which means you can now use smaller particle size columns to really improve productivity and get faster run times. It also offers enhanced temperature control, which you do need for haloacetic acids. They are temperature sensitive, so we've recognized that fact, and we do have this enhanced temperature control component built into the system. If we look at the X-ray view of the system, you can see that System 1 consists of a pump, as well as the standard EGC. We have a System 1 detector and a System 1 column compartment. The Autosampler is actually common to both systems, and it actually works with both systems in conjunction. System 2, we have a capillary-based pump so that we get that 4 millimeter down to 0.4 millimeter drop. And we also have a low temperature DC to accommodate the temperature requirements of the methodology, as well as a System 2 capillary EGC, which allows us to do element generation on the second dimension as well. So what does the chromatography look like? Really, this is where the rubber meets the road with this information. And you can see here we're doing a first dimension separation on an AS24. This happens to be a sample of 20 ppb of the HAA9 in a high ionic strength water, this in the upper chromatogram, and a 100 ppm ammonium chloride solution in the lower chromatogram. So in the absence of the second dimension, you can see these peaks would actually be very difficult to quantitate. There's a lot of peaks that are actually on the shoulder of the larger peaks, which makes quantitation nearly impossible. And so this, again, in the first dimension, if we were trying to quantitate something in the first dimension, would be next to impossible. If we do now a look at the second dimension, you can see that same chromatography now processed through the second dimension of the system, and the peaks are much better, they're much sharper, they're much easier to integrate, which is ideal for anyone that's trying to quantitate the haloacetic acids. Here's actually the lowest concentration method reportable levels for the HAAs relative to the 2D-IC and method 557. So you can see the 557 is a little bit more sensitive in some areas, but then the 2D-IC method is actually sensitive in some areas as well. So they're quite comparable. If we look at some of the haloacetic acids in groundwater, you can see this is actually a second dimension separation of a groundwater sample, and then that sample A spiked with 10 micrograms per liter of the HAA9. And so we're seeing elimination completely of the groundwater matrix and nice quantitation possible of the haloacetic acids in groundwater. If we actually look at a surface water, again, similar results. Now the run times for the HAA analysis are going to be long, which is in some cases might be a detriment, but since the system is fully automated, you can actually run the HAA samples overnight or in an unattended operation, which makes it ideal for unattended analysis. Here's actually some recovery results that we see for the haloacetic acids. This is the HAA5, good recovery, as you can see, in both the surface water and the groundwaters. And we also do some comparison in this table showing the 2D-IC results and comparing that to 552.3. And so the detection limits relative to 552.3 are actually quite good. In fact, in 552.3 for the MBAA, we weren't able to see any of the results, but with 2D-IC, we're actually able to see and detect and quantitate that compound. So in conclusion, 2D-IC is an ideal method for the haloacetic acids because it's sensitive and it's selective. It's going to be a wonderful alternative to 552.3 because it doesn't involve the lengthy or laborious sample preparation steps that we talked about that are part of the GC ECD application. So with this, this technology, HAAs can be directly determined. There's no sample preparation. It's simply loaded onto your Autosampler, inject it, and you're ready to go. It's selective, it's sensitive, and it's designed to eliminate the matrix associated with typical groundwater samples. So in a situation where you have drinking water, you have groundwater, surface waters, the first dimension is there to eliminate that matrix. The second dimension is there to really optimize the chromatography. Similar to what we have seen with bromate as well as perchlorate, 2D-IC is really a nicely optimized technique that has general acceptance in the industry. In fact, the EPA has approved it for bromate as well as perchlorate, and we definitely anticipate that the haloacetic acid method is well underway as well. So it's simplified, it's less costly, it doesn't involve additional detectors like mass specs or triple quads. With the enhanced temperature controls, it really is ideal for doing two-dimensional haloacetic acid. That's it for my presentation on haloacetic acids using two-dimensional ion chromatography. Thank you so much for your attention, and if you have any questions, there's plenty of resources. Please reach out to us and we'll be happy to answer those for you. Thank you.